OSC New Tech. And as always, a very big thank you to all our sponsors who made this wonderful podcast series possible. I want to let you know that this episode is being recorded and you will be able to look at it on demand from the All Single Cell website, where you can also find the recordings from the previous episodes. So let's get started. We have three brilliant speakers here today: Professor Miguel Esteban, Dr. Rachel Thijssen, and Dr. Mike Clarke. And each one of them is ready to talk to you about their favorite new technology and their new approaches that allow them to push the boundaries of their research. So I want to start by introducing our first speaker, Professor Miguel Esteban. He is a Spanish physician and scientist fully based in China. He did his postdoctoral training in cancer biology in the Imperial College Lab of Patrick Maxwell in London. He is now a professor at the Guangzhou Institute of Biomedicine and Health since 2008 and a scientific advisor at BGI Research since 2019. His research focuses on unveiling the transcriptional and post-transcriptional regulatory mechanisms controlling cell fate transitions. And together with his colleagues at BGI Research, he is creating a comprehensive molecular atlas of human disease and body regeneration using single-cell omics technologies. The title of his talk today is Stereomics, Spatially Resolved Transcriptomics at 500 nanometer resolution. And with no further ado, over to you, Miguel. Please share your screen.
Hello? I'm not sure I can get started.
We can hear you, Miguel. Off you go. Thank you.
Okay, thank you. Sorry about that. First of all, Joseph and Daniela, thank you very much for the invitation. It's a pleasure to participate in this webinar. So my name is Miguel Esteban, and I'm a professor at the Guangzhou Institute of Biomedicine and Health in Guangzhou, China. And I'm also scientific advisor at BGI Research in Shenzhen. The title of my talk, like Daniela said, is Stereomics, Especially Resolved Transcriptomics at 500 nanometer resolution. Okay, so just a little bit of a very normal introduction. So we all know that tissues are heterogeneous, and if we really want to understand how normal physiology and disease works, we cannot just study average measurements, and we need to get at the single cell level. And if not, we're going to miss rare effects, and we're not going to be able to understand how what things work in normality, or how things can go wrong. So we are experiencing now a revolution, a single cell revolution, and the same way that Manhattan of the 19th century through the Industrial Revolution became what we know this big city is. Our views of cells and tissues, thanks to these technologies and other we cells and tissues, and we so to a digital people. Technologies. We to and
Miguel, Joseph here. Your sound's not coming through, but the videos are playing. Can I suggest that you maybe turn off your personal video, so it might save some bandwidth? Miguel, I'm afraid we still not getting. I'm not getting any audio through. We're getting a few messages from participants that we can't hear you. Maybe it's worth just taking, just checking your audio settings. If not, we can all use our imaginations and imagine what you're talking about. All right, so it looks like Miguel is just reconnecting. Sorry about the AV issues, everyone that's listening online. I seem to be cursed. The last 2 episodes I've been involved in have AV issues, so hopefully we're back on track now. Let's have a go. Miguel, I'm afraid we're still not getting your audio coming through. Could I maybe suggest that you that we move on to Rachel and have Rachel speak next, and just you can investigate if there's any possibilities of connecting through a different audio system. I think that's come through. So sorry everyone about the change in the slight change in plans there. So hopefully Miguel will be able to connect and we'll be able to hear about his work. It looks fascinating based on the slides I can see at least. So we might just have a little change to the schedule and move forward and put Rachel on the spot to, or not to present. Miguel, could you stop sharing your slides and maybe we'll let Rachel speak and then come back to you later on if that's okay. So in that case, I might just be a bit ad hoc and just introduce Rachel. Rachel Thijssen is a senior postdoc at the Walter and Eliza Hall Institute of Medical Research. She completed her PhD in hematology department at the Academic Medical Center in Amsterdam, the Netherlands, before relocating to Australia. And she's going to be talking to us today about her work on how blood cancer cells evade death, and particularly elucidating the emergence of the resistance of venetoclax-treated CLL using novel single-cell multi-omics approaches. So Rachel, thank you very much for jumping order, and take it away. And hopefully your audio is working.
Yeah, so I hope everyone can hear me, and otherwise shout out.
We can.
So, yeah, today I will be talking about a novel multi-omics approach, and I will just give a very brief introduction about what CLL is and why we need to develop this multi-omic approach to answer clinical relevant questions. So CLL stands for chronic lymphocytic leukemia, and it's the most adult leukemia in the Western countries. And every CLL cell and every patient overexpress a pro-survival protein called BCL2. And research done here at the WEI led to a development of a small molecule inhibitor called venetoclax. And the first CLL patient in the world was treated here in Australia in 2011 with this drug called venetoclax. And the first clinical trials showed a really good success rate, like 80% of these patients responded to venetoclax. And this is just one of the patients that was treated with venetoclax here at the Royal Melbourne Hospital. So you can see that this patient had very bulky disease in the lymph nodes, so a lot of tumor cells in the lymph nodes. And just after a couple of months on venetoclax, this disease was completely gone. So the lessons that we learned from the first in-human clinical trial is that we know if you target BCL2, it kills these tumor cells very rapidly. And we saw a response in all compartments. So if you take samples from the peripheral blood, the lymph node, or the bone marrow, we could see a leukemic burden going down just in a couple of months on venetoclax treatment. So after these clinical successes, venetoclax is now being approved worldwide to treat high-risk CLL, but also here in Australia, it is listed on the Pharmaceutical Benefits Scheme. However, venetoclax is not the cure for CLL. So even though most patients respond really well to venetoclax, and you can see that the disease is completely gone, after years of venetoclax therapy, you can see that in some patients, the disease is coming back. And if you just look at the first in-human clinical trial, around 80% of these patients, the disease is coming back. So the big question, of course, is what's happening in these CLL cells? So why can they escape killing by venetoclax? And to investigate this, we first, of course, looked at its target itself, so BCL2. And we did this with conventional methods like digital droplet PCR and whole exome sequencing. And we looked at screen versus relapsed patient samples. And what we found was a mutation in exon 2 of BCL2. And this mutation then changes the glycine on the 1-on-1 residue into a valine. And it changes the structure of BCL2 so much that there's less venetoclax affinity to this mutant BCL2. And we can model this, and we showed, of course, that cells that harbor this BCL2 mutation are less sensitive to venetoclax in vitro. So this is great, but it is a much more complex story. So not all patients that relapse on venetoclax harbor this BCL2 G101V mutation. And in the 40% that does have this mutation, it is subclonal. So if you look, for instance, at the CLL compartment depicted in this gray round, and then look at how many CLL cells harbor this mutation depicted in blue, you can already see that it's very subclonal. So the big question, of course, is what's happening in the other cells that then also could escape killing by venetoclax? And to answer this, we want to, of course, apply single cell technologies to uncover why venetoclax fails. And working with patient samples, and I think a lot of researchers can relate, we're very limited to the amount of samples that we got. So we want to get as much information out of a single sample as possible. So therefore, we want to apply multi-omic approaches. And over the years, there are many multi-omic approaches available. For instance, looking at the genome together with the transcriptome, or the transcriptome together with the proteome. And I'm not going to go into it, but what we're interested in is combining transcriptome sequencing with proteome. And this is great because then you can do it via 10x genomics. And we can process up to 10,000 cells per sample. The only problem with 10x genomics is that it's reliant on short-read sequencing. And it's great to look at gene expression, but it then loses the isoform detection or maybe SNP variants to call in those short-read sequencing. And we know that gene function can differ based on isoform. And we also know that isoform usage in cell can vary based on diseases such as cancer. So not to only perform short-read sequencing, what we also want to do is long-read sequencing. And if we do long-read sequencing, we not only get gene expression, but we can also detect isoform usage. And we can even call SNPs in these long-read sequences and hopefully maybe even detect a BCL2G1R V mutation. The only problem is that long-read sequencing is still relatively expensive. And to overcome this, Louis Tian and Jafar Jarabi developed a new method called single-cell full-length transcriptome sequencing, or FLIT-seq. And you can reduce the cost by two ways. Or subsampling the cells, so you have less input of less cells. In the end, you need less reads, of course, or more targeted approach, so looking at specific genes. So this is just a 10x workflow scheme, and I think many of you already know this scheme, so I'm not going to go into it that much. But doing FLIT-seq, you can actually do it, you can target it in two ways. You can do subsampling, so if you form the gems, you can split it in two, and only from a small part, you can do full-length transcriptome sequencing. Or when you have the 10x cDNA, or that's already barcoded, and it's full length, then you can come up with a targeted approach. And we developed a new method for that as well. And I will discuss this later in my talk. To analyze all this data, Louis developed a new data analyst pipeline called FLAMES. And what it does is just still processing the short-read sequencing as normal, according to the standard 10x procedure, and also clustering these cells based on a 3 or 5' expression analysis. And then it uses a cell barcodes from the short-read sequencing to demultiplex the long reads and then overlay this on this short-read clustering analysis. Also with this method, we can also integrate other omics data, such as protein expression. So for this project, this is the single-cell multi-omics approach that we undertook. So we have CLL patients before and after they relapsed on venetoclax. We took the peripheral blood PBMCs and we stained them with a surface protein marker panel from Total Sexy from BioLegend. Then we performed 10x. And after we have these gems, we split them into 80% and 20%. And we treated it as two separate samples. Of all the samples, we prepared transcriptome libraries and proteome libraries with the Illumina library kit, and then sequenced on the next stick. And of the 20%, we also prepared whole transcriptome nanopore libraries and sequenced it on the Promethean. And we also on the 80% had a targeted approach, what I'll come back to in a minute. So this is just the data from one patient that relapsed on venetoclax. And we're looking at the UMAP on the short reads sequencing results. So what you can see is that looking at short reads transcriptome, you can see that the monocytes cluster nicely together, the T and NK cells cluster really nicely together, and you see a big cluster of course, the tumor cells. What you maybe already can appreciate is that within the tumor cells, we see already subclusters. So it's not a heterogeneous population. If you then look at the short reads and long reads, you see that there's an excellent overlap between the short reads and long reads. So that's great. So then we're going to investigate this long read sequencing. And very lucky, we could detect the BCL2G1R V mutation. And what you can see is we can detect the BCL2G1R V mutation in the FLIT-seq data, and it clusters mostly on cluster 1, 2, and 6. However, the coverage is not great or optimal, And therefore, we developed a more targeted approach to perform it on the rest of the 80% of the cells. And to do this, we developed a new method called rapid capture hybridization sequencing, or short RAGE-seq, that we performed on the 80%. And this method, what we did is we designed probes of our genes of interest. And these probes are 120 base pair long. And we designed it to every 1 kb exon. So if the exons are bigger, we design 2 probes. If the exons are smaller, we align them and design a probe to that. So in the end, for our 14 genes, including also BCL2, we got 293 probes. And if you then look at, for instance, the BCL2 G111 coverage, you can see with short read sequencing, we don't have any coverage. What it's expecting because it's short reads. With the full transcriptome library of Nanopore, we have some coverage of BCL2 G111V. But now with our specific capture method, we can see we have very nice coverage. So this is great. So now we can look what's happening in the other cells that don't have G111V. So first, just to show here the RAGE-seq data. So again, CLL2, and these are all the CLL cells. And now you can really see that in cluster 1 and 2, you have the most cells expressing this G111V mutation, also depicted here in this bar graph. Now, really knowing this, that's mostly in those clusters. Now we can look what's happening in the other cells. So then again, we can also go back to the short read data, of course, and look at gene expression. So on long reads, we can see G111V, and based on short read data, we actually found a very interesting gene being upregulated, BCL-XL. So this is another pro-survivor gene that is normally not expressed by CLL cells. And what you can see is that this gene is mostly expressed in a different cluster that's mutually exclusive of the G111V. And this gene is not targeted by BCL2. So it's another resistant mechanism that happens at the same time in these CLL patients together with this mutation. Also, by performing full-length transcriptome sequencing, we can also now characterize the isoform usage. And you can then map it, of course, to a reference. So if you have a full splice match, it will just be the same as the reference. You can have novel splice matches that are in catalog or novel that are never been reported before. And looking at CLL2, what you already can see is that most of the T cells have a full splice match, so we can map it back to the reference. But all the tumor cells, what you already can maybe appreciate, depicting more in the blue colors, they don't have a full splice match. And then if you look at the novelty, you actually can see that most of these CLL cells express isoforms that you can't find back in the catalog. And why is this now supported to look at isoforms usage in these cells? Because when we looked at a specific gene, I'm just going to call it gene X in this regard. If we did just single cell RNA-seq and look at the short read sequencing, it seems that all the cells express this gene normally. And also based on full-length transcriptome, most of the cells express the normal transcript. However, we found in the FLIT-seq data a very small cluster in cluster 4 that has this novel transcript or novel isoform, and that's very never been detected before. And we also find it back in the RAGE-seq. So also it's showing that targeted approach can also detect novel isoforms. What's more interesting, if when we look at cells when we make cells that express the normal transcript, these cells are very sensitive to venetoclax, but when these cells express this novel isoform, they become resistant to venetoclax. And this is really just showing and highlighting how important this long read sequencing is, because we didn't detect it just by short read sequencing, and we also didn't detect it by the bulk sequencing as well. So just to summarize this talk, so our novel single-cell multi-omic approach reveals unexpected and complex heterogeneity in CLL cells relapse on venetoclax. So by doing just short read sequencing, we already can find subclusters that have a different gene expression. And by now combining it with full-length transcriptome and more targeted approach like RAGE-seq, we can also detect mutations, and we can also detect novel isoforms. And we also overlaid it with protein, but I didn't have time to show this today. So, and then my last slide, of course, before I forget, the acknowledgements. So I really want to thank Louis Etienne from the WEHI here and Jafar Jawi from AGRF my supervisors, Andrew Roberts and David Wang, and the funding bodies that makes this work possible. So thank you.
Rachel, thank you very much. That was absolutely fascinating. I've got a lot of questions and there's a couple filtering through the chat, but we'll, in the interest of time and also to give Miguel an opportunity to reconnect, we'll hold off the questions to the end as in a panel discussion. But just very quickly, did you name that targeted capture approach after yourself?
Yes. So actually someone else came up with the name, Stephen Wilcox from Lehigh. And then I was like, yeah, why not? It's probably the first and last time I can name something after myself.
Yeah, this is fantastic. It reminds me of Victorian naturalists naming animal species after themselves. Great. So thank you, Rachel. We'll move on and invite Mike to speak next, if Mike is happy with a little bit of change in the order while we're waiting for Miguel to come back on. Mike, are you there? Is that good for you?
Yep, that should be fine.
Brilliant. All right. Well, so I'll just introduce Dr. Mike Clarke, who is a group leader in the Centre for Systems Biology at the University of Melbourne. And he started a lab, was it about 1.5 years, 2 years ago?
Yep.
And is making some fantastic progress of bringing together a range of new technology approaches to solve some pretty interesting biological questions. So, Mike, over to you to give your presentation. Thanks very much.
Okay, can everyone see that all right? All right, well, thanks very much, Joseph, for both the invitation and the introduction. So I'm going to talk today a little bit about using targeted single cell sequencing specifically for transcription factors to look at cell identity and cell differentiation trajectories, some of which has been about the capture targeting has been nicely introduced by Rachel just before. So just as a bit of an overview, I'm going to mention briefly about why you might want to use targeted single cell sequencing to help overcome issues with sensitivity and variability, how we've used it to look at transcription factors as a pool to investigate our stem cell differentiation into cortical neurons, and how we think potentially targeting of transcription factors might be quite an effective way forward for examining both cell types and differentiation. And this work is currently under review. So, as I'm sure we all know, one of the great strengths of single cell sequencing is you can profile gene expression in single cells, and that's been used extensively to identify cell types and their expression profiles. An example here, but I could have picked any one of hundreds of papers. And also, it can create some really beautiful cell differentiation and disease trajectory work. Again, some great work here on zebrafish development. One of the sort of known weaknesses of scRNA-seq is that the sensitivity compared to bulk sequencing is lower, and you tend to get high variability. And this can mean that many expressed genes are missed. And I guess sometimes what we might ask is, what biological information does this mean we are now missing? We thought a while back that using RNA capture-seq, otherwise known as targeted sequencing, might be at least a potential solution to some of this low sensitivity in scRNA-seq. As sort of introduced by Rachel, capture-seq targets a portion of the transcriptome for focus sequencing, and this gives you a really huge increase in your sequencing depth and your coverage of your genes of interest. And the way it tends to work is you design probes against regions of interest, and then you mix those probes with your the library you've made before sequencing. There'll be hybridization between the probes and your target genes. You can then pull those out, wash everything else away, and really focus your sequencing on your genes, isoforms, genomic regions of interest. And then allows you to quantify genes and isoforms, and in this case, would hopefully enhance your profiling of expression between cells. So we know from bulk sequencing, this produces a huge increase in sequencing sensitivity, and a few years ago, we wondered if it would do the same in single cells. There's now been a few studies showing that this definitely works in single cells and works quite well. Usually applied to sort of smaller numbers of genes. So like, as in the work that Rachel was showing, which looked really beautiful, and RAGE-seq from the Garvin. And so we wondered whether if we targeted the often quite lowly expressed transcription factors, whether this might help us improve cell type identification and differentiation trajectories. And so the model we decided to apply this to was stem cell to cortical neuron differentiation. So this is a 90-day cell culture process where you start with embryonic or induced pluripotent stem cells. They go through a neural induction into neural progenitors, and then slowly over time you get neurogenesis for deep layer cortical neurons, upper layer cortical neurons, as well as some astrocytes. And this is basically a process that temporally recapitulates development in humans. And what it's been validated quite a few times to do is generate cortical excitatory neurons. It's been a really useful model for trying to help understand brain development and also to look at brain disorders. And some of our collaborators in Oxford, when I was there, started a big project doing this, and they wanted to understand, how robust this method was. And so they had 5 different labs. They took 2 different cell lines, and they all had a go at making these neurons in this sort of 90-day procedure. And then they took a couple of time points. This says FP, that's after final plating here. So this is really like day 50, and this is really day sort of 80, 90. And then they compared how well it worked using bulk RNA-seq and proteomics, etc. So the good news was that everyone made neurons, shown here in green for a couple of classic neuronal markers. But what their bulk RNA-seq also showed was that samples all seemed to separate by lab, not by cell line. And that was obviously not what they were hoping for, sort of huge lab inter-lab variability. And so they decided to do some single cell sequencing on a couple of the samples from a couple of the labs to try and understand what had happened. Why were the neurons essentially from each lab looking so different? And using some standard single cell RNA-seq, they concluded that what had happened was that the labs had created different ratios of neurons on glia. And so that was kind of our starting point for this project, because what we wondered was, could we do a better job of understanding what had gone on here? And if we targeted transcription factors instead of the whole transcriptome, could we better understand this neural differentiation and the causes of the variability in this process between the labs? And to do this, we targeted all transcription factors with our capture. So that's over 1,000. You can definitely do this. And we did this on the sort of leftover single cell libraries that had been sequenced previously, and then we repeated the single cell sequencing. So now we had matched, before standard single cell sequencing and then post-capture single cell sequencing for each cell. Because this was started a couple of years ago, this was done on SmartSeq2 libraries. It would be just as applicable to 10x and many other types of library prep processes. And to keep things fair, we only analysed the cells that had passed the QC and standard scRNA-seq. So that was about 300 cells, which isn't so many these days, but it was more than sufficient to answer the questions we were interested in. So technically, we found that the capture dramatically improved transcription factor detection. So we went from about 2% of reads pre-capture being on transcription factors up to around 75 or 80%. So that was about a 36-fold enrichment for transcription factor reads. We were able to detect about an extra 150 transcription factors, which were not seen at all in any of the cells pre-capture. And we also detected a lot more transcription factors per cell. And so our hope was this would allow us to better identify kind of exactly what cells we were looking at. So what sort of cell types were there? Okay, so these were the pre-capture results based on the previous study, Volpato in 2018. When we took their data and sort of used UMAT and put out the clusters, we got the same as what they did. We saw essentially 4 clusters. One in green here looked like glial cells. This came from both labs. And then one lab's neurons were found in these 2 clusters here, and then the other lab's neurons were found in 2 clusters here. And the previous conclusions have been, these look like astrocytes, and there was variability in the ratio of neurons to astrocytes from each lab. So we did our capture sequencing, and we did the same thing with our clustering. We found 3 clusters, so G, N1, and N2. These are shown here. And then here are post-capture clusters, but they're colored by pre-capture, so they're more comparable. The first thing we found was that this supposed glial cluster was not glia at all. They were not astrocytes. They were in fact radial glia, which are neural progenitors. So these are the neural progenitors you generate in this differentiation process that then will go on to become the neurons that you generate. And these were found in this G cluster here. Both labs made these. Lab E, this N1 cluster here, or this what was formerly these clusters 3 and 4, these were excitatory cortical neurons. That's exactly what we'd be hoping to find. So that was all good. The biggest surprise was that this cluster here, which is N2, were not cortical neurons at all. They were inhibitory interneurons. This is not what we'd expect this procedure to generate, and is not what we were able to detect previously. But if we look at the differential expression of transcription factors between these 2 neuronal clusters, what we can find is that this cluster N1 expresses a large number of transcription factors, which are indicative of excitatory cortical neuron fate. And then in cluster N2, we see a lot of interneuron markers, including SP9, etc. So really classic transcription factors for interneuron fate. If we had a look at this pre-capture using these same cells, there was, using the pre-capture libraries, there were some differences, but that kind of cortical neuron interneuron cell type difference really didn't show up strongly in the data. So what we conclude from this is that unlike what the pre-capture data showed, or standard single-cell RNA-seq showed, differentiation into different types of neurons actually explained the differences between the labs. And this suggests that targeting transcription factors can really actually enhance and potentially correct cell type identification in scRNA-seq. Because interneuron identity was quite unexpected, we actually wanted to validate that was really what was going on. So we went back to the lab and said, by the way, we think you're making interneurons. Can you check this? And so they took some later cultures and had a look at this. And what they could find was some of their neurons were expressing GAD65, which is a marker of inhibitory interneurons. And then when they did patch clap electrophys of these neurons, they found that you could block synaptic currents with picrotoxin, which is again consistent with GABA inhibitory interneurons. So we're pretty confident that we got that one correct. We also had a look at how you can use capture of transcription factors to look at developmental trajectories. And so this is using slingshot pseudotime trajectories. So on the left here, we can see the pseudotime by cluster, and on the right here, we can see it actually by pseudotime, where sort of the brighter yellow here is early and the darker is late. And what we can see is that the earliest cells in the trajectory are in fact that G cluster, which supports their radial glia progenitor identity. And the other thing we sort of noticed was that even at the very early stages, the radial glial cells in the 2 different labs seem to kind of seem to have already separated. They seem to have a fairly different trajectory from an early stage. And so we wondered if we could work that out, why that was. And so we did quite a lot of work building gene regulatory networks and so on, and looking at protein-protein interactions of these genes. And to cut a bit of a long story short, what we found was quite a high enrichment for transcription factors involved in retinoic acid signaling. And we found that these negatively correlated with interneuron transcription factors. And you can sort of see this here. Here we have the lab D, so radial glia to interneuron cells sorted by pseudotime. And then here we have lab E, so radial glia to cortical neurons. And what we find is that many of the transcription factors to do with retinoic acid signaling, so RORA, for example, here, or NR1H2, we see them sort of sometimes early in the lab D, but then they all tend to get switched off, whereas that doesn't happen in the lab E samples, which became cortical neurons. So it looks like to us that there's an early cessation of retinoic acid signaling that may have been the switch leading to interneuron fate. And it's worth pointing out that retinoic acid signaling is known to be important in brain and cortical development, and actually having retinoic acid signaling is part of the procedure to generate the cortical neurons. So it looked like there'd been kind of perhaps a failure of that in the lab D, which is why they were generating interneurons. So basically, to summarize what we were able to find, we think that using capture-seq targeted RNA sequencing provides improved gene expression profiling in single cells. We think that using the capture of transcription factors specifically can really improve your ability to identify cell types and differentiation trajectories. And that in our case, doing this really clarified the neuronal cell types that were present and identified a developmental switch in stem cell to neuron differentiation. When we think about this methodologically, we perform this with SmartSeq2 libraries, but this would be completely applicable to 10x. Basically, anytime you've got a prep where you can got your library that's basically ready to go for sequencing, you can, if you want, basically do your capture on that, or at least a portion of that, and then you can also see, if you want, sequence a captured or targeted sequencing library. Most of these advantages I've been talking about are about increased sensitivity, but one of the other advantages of performing targeted RNA sequencing is your ability to massively increase your multiplexing without giving up any read depth. So I'd just like to say thanks to everyone in my lab. This is all of them here. It's been a bit of a crazy year. There are people in my lab who I've actually never met in person, but hopefully that will change soon. And just thank everyone who's been involved in some of these projects. This specifically has been with Zam, Adam, and Alex at Oxford University, and all our funding agencies. And thanks everyone for listening.
Mike, thank you very much. Yeah, again, that was absolutely fascinating. I really enjoyed the intersection of using the sort of targeted capture and those alternative read technologies to resolve some non-trivial issues that you have with differentiation of stem cells. We will also hold off questions until the end in the interest of time, but I'd like to just apologize. I introduced the centre that you belong to wrong at the beginning. I'm going to blame it on an extremely early start that I had this morning presenting at a webinar myself at 2 a.m. So Mike is at the, I'm going to just double check this before I say it, is at the Centre for Stem Cell Systems.
That's correct, yes.
So apologies.
Lucky, I think Christine is also presenting somewhere else, so she's not here to
oh, right. Well, that's good. I was mainly apologising because I thought I was going to get in trouble from Christine. So,
yeah.
Yeah, so thank you very much. So Miguel has migrated to a different room in his institute, and hopefully a room which is closer to a Wi-Fi router. So we're going to try for Miguel to start his presentation again. We've asked us to start from the beginning. Miguel, if you want to share your slides, and we'll just see how the audio comes through. Thank you.
Okay, so can you hear me better now?
Yes.
Yes.
That sounds good.
That's a relief. Okay, so I'll get started. Again, thank you very much, Joseph and Daniela, for the kind invitation. And I apologize to the entire audience for this technical problem. My name is Miguel Esteban, and I work at the Guangzhou Institute of Biomedicine and Health in Guangzhou, southern China. And I'm also a scientific advisor at BGI Research in Shenzhen, in also southern China, near Guangzhou. Okay, so I think I don't need to talk too much about the heterogeneity of tissues and the idea that if we only get average measurements, then we cannot understand normal physiology and disease. So thanks to the evolving single cell sequencing technologies that we are experiencing, this single cell revolution, the same way that the Manhattan of the 19th century became transformed into the Manhattan that we know today, our view of cells and tissues is being transformed from a prehistoric concepts to digital concepts, so that we are having the digital cells, and we are heading towards the digital human. But single cell technologies, like we have been hearing, now, and we all know, have a series of limitations. And very importantly, is critical to balance cost, throughput, and sensitivity, because in the different approaches that are available, the three things don't go simultaneously most of the times. So how can we do this? How can we balance cost, throughput, and sensitivity? So with my colleagues at BGI, we have been thinking how to solve this issue, and we have created this DNM-B Lab C4 technology, which is disposable, portable, and cost-effective microfluidic system. And the principle is very simple. We can see a video here in which, okay, I'll wait a couple of seconds. So the solution is loaded on one side, and then the physical principle for the whole instrument to work is negative pressure to a syringe. And then this makes the whole creation of libraries very cost affordable, and using MGI sequences, the whole process it can be really, really affordable. Oops. So we're using DNM-B Lab C4 based technologies for different purposes. One is the single cell RNA-seq. Another one is single cell ATAC-seq. And more recently, we have also developed a multi-omics approach in which we can combine both single cell RNA-seq and single cell ATAC. And we are increasing the throughput of this C4 system, with the goal that ultimately we would be able to achieve hundreds of thousands of cells in one row for both RNA-seq and ATAC-seq. And we are developing more applications of the C4. Single cell RNA, nascent RNA sequencing is one of those that are coming out very soon. And we welcome new ideas, and we are very happy to work with anybody from all over the world on trying to create new applications. This is one of the first papers that came out using the C4 technology, is profiling of blood from patients with COVID-19. And another project that we have just completed is the single cell transcriptome landscape of a non-human primate, Macaca fascicularis. So we have profiled 43 organs from 8 major systems, and the total number of nuclei that we have sequenced, nuclei or cells depending on the tissue, is over 1 million cells. And we have been, thanks to the depth of the system, that we can get basically with tissues, we can get anything around 1,500 to 2,000 genes per cell on average, sometimes more. And with cell lines, obviously, we can go very, very, very high. And thanks to this, we have been able to study different signaling pathways in the that regulate the physiology of a non-human primate. But what comes next? So next, obviously, is one of the very vibrant technologies that are emerging, is the spatially resolved technologies, because even though you can study tissue heterogeneity using single cell omics, you cannot understand the position of the different cells. And without understanding the position, you don't really understand how the tissue or the disease is really working. So we have a series of very interesting technologies that have appeared over recent years: Visium from Tenex, Slide-seq, HDST, and more recently BIT-seq and others. And so they are very interesting, but the future is heading onto the spot size that they use being smaller, the pitch size being denser, and the field of view being wider. And also, I would like to add that the number of genes that can be captured are bigger, that are somehow related to some of these parameters. And thanks to spatially resolved technologies, we would be able to understand the panoramic view of single cell omics, and then through these, by capturing function and structure for the cells in the context of a tissue, we'll have very significant breakthroughs in the understanding of life. So, oops. Okay, so my colleagues at BGI had a very good idea, which is then the sequencing systems, NGI sequencing systems, are based on DNA nanoballs. Basically, the DNA is amplified and using this technology, and then they are placed on pattern arrays, and this is a principle of their sequencing technology. And they had the idea that maybe this can be used for special transcriptomics. And we can see here that if you place the DNA nanoballs in these gaps, then you can put poly-T sequences so also attached to these DNA nanoballs. And then the idea was to place tissue on the top of these slides, and then if we could capture the RNA and the RNA is transforming DNA, then we would be able, because these DNA nanoballs are barcoded, we would be able to get an idea of the distribution of all these cells or these spots in the tissue. So one of the first approaches that we have used with this technology that is called stereoseq, and I would like to say that we can the resolution that we can get is very high. We can get down to 500 nanometers to 715 nanometers. So we're moving within this range, and we can capture a good number of genes now per 100. So basically, basically we can have if you imagine that cells have around 10 to 20, 25, depends on the cells and depends on the organism of size, then we can have hundreds of DNA nanoballs per cell. And then per 100 nanometers beam, we can capture 8,000 genes so far, and we believe this can be improved much more. So one of the first approaches that we have been using this technology that we call stereoseq is to profile tumors. We have here a sample of liver cancer, and you can see that there's different regions can be captured through our stereoseq technology, and then we can see here the different terms that appear based on the genes that we have been capturing, and we think that this is going to be able, since we can profile this systematically and we can do many slices simultaneously, we think that this would be able to advance very significantly, not just our understanding of cancer at the molecular level, but also diagnostics, because we hope that we can bring this to the clinic. This is another proof of principle of the stereoseq technology, in which we have aimed to reconstruct the mouse olfactory bulb, especially, and we can see here the tapping. This is the UMIs that we can capture, and this is the clustering of the predicted cell types based on the genes that we have been identifying. And then we can see here that for specific genes, when we compare to the Allen Institute brain map, we can see that the distribution of specific genes that are markers of different zones of the olfactory bulb, the similarity is really quite striking. So we have also here for ganglion cells, we have another gene, the PCP4. So it's very striking. And since we can capture we have many different nanoballs per cell, we can capture transcriptomics in a spatially resolved manner at a subcellular level. And this is very important because tissues are complex, not just in the composition, but also in the way the different parts of the cell gets into the tissue. So for example, for neurons, for the synapses, and then not just for those very specialized cells, but for others. So thanks to our technology, we can get into understanding how different domains of the cells have different transcripts in an spatially resolved manner. And we can see here intronic reads in the nuclei that can be captured using our approach. This is a mouse brain section, also proof of principle of our technology, and you can see the clustering that it can visualize the different anatomical areas, and these are this is the hippocampus, and then these are some marker genes of the hippocampus like HPCA. And then what we did here is try to see whether we can detect the difference between the soma of the neuron and the axon that go into this direction. And here, through computer analysis, we can see the peak of a gene that is mostly expressed in the soma, and then these other genes that are related to the synaptic function and others, then mostly in the axon. So another proof of principle and a very relevant approach that we have been using is studying mouse embryogenesis with the stereoseq technology. This is hematoxylin and eosin of a mouse. I think this is 16.5 or 17.5, and you can see here the clustering and the predicted regions here, and you can see that the striking similarity between the regions in the staining and the regions that we detect with our stereoseq technology. And these are just some marker genes for different tissues that we have been testing: albumin in the liver, muscle, LACTA1, so many others. And here is mouse embryos of different stages. We have 8.5, 11.5, and 16.5. And the idea of this was to try to see whether we can study brain development, central nervous system development in an especially resolved manner using our technology. These are the UMRs, and this is one of these genes involved in the central nervous system development. As you can see, at a very early stage, starts being enriched here, then it goes through the whole spinal cord, and then finally, so we also have signal here, but it's mostly is enriched here in the part of the developing brain. So a little bit of a summary of our stereoseq technology. Thanks to being able to have multiple DNA nanoballs per cell, and these nanoballs being able to capture different genes. So we have a very high resolution, and we have a very high sensitivity. Like I said before, per being of 100 micrometers, we are on the scale of around 8,000 genes now, but we think this can be improved very, very significantly. And it also has high scalability in that now we have been using first smaller chips for the mouse brain, but we are moving into much larger chips so that we could do not just on one slide the mouse brain, but we could do the mouse brain that we're doing already, and even in the human brain, which is around 13 centimeters of diameter. And this is very important because the same way that there's batch effects between libraries of single-cell RNA sequencing of cells on nuclei in suspension, so for studying tissues, if we can have the whole tissue on the same chip, we would be able really to understand patterns of disease or normal physiology function with much higher level of accuracy, especially when we are comparing samples. And I would like to say regarding the scalability that my presentation today is about RNA sequencing in a spatially resolved manner, but we are already moving into ATAC sequencing in a spatially resolved manner using this principle, and then we predict that that at some point we'll be able to also do it multiomics at a spatially resolved in a spatially resolved manner, and also potentially other type of sequencing technologies such as whole genome sequencing or Hi-C. So one of the projects that we are working on now is single-cell multiomics atlas of mammalian development. We're using our C4 technology for multiomics so that we can have in the same cells the single-cell RNA-seq and the single-cell ATAC-seq with the cells in suspension. And we are also using the spatial transcriptomics, and we're doing this with a whole range of species going from the mouse to the human, and with emphasis on non-human primates, different types of non-human primates. And I think we believe this is going to be very important for trying to understand the principles that differentiate the different species, and hopefully we'll be able to come up with new ways of reactivating those principles, those molecular machines and genes in vivo, so that we can foster regeneration in humans. And then I would like to say that this work and other works that I presented before, like the Macaca fascicularis single cell atlas, have been done in the context of a single cell consortium that we have established in southern China. It's around the Great Bay Area, Shenzhen, Guangzhou, Foshan, other cities, but we are expanding to other cities and institutes in China. We already also have collaborators from all over the world, and the idea is to sequence over the course of the next few years billions of cells, and these would be achieved not just thanks to the progressively larger scale of our C4 technology, but also considering that especially resolved transcriptomics allow to sequence many cells at the same time. We think that we are going to be able to create very comprehensive maps of human and mammalian life in general, and actually we're going also to other animal models. And finally, I would like to say thanks to all my friends and colleagues from BGI Research, Xixun, Hoyong, Chen Ao, who is the mastermind behind all these especially resolved technology that I've been mentioning. Of course, Liu Longqi and Xiping, and then many other colleagues I'm not mentioning. And this is Chen Ao's team. And these are my colleagues I've been working with for the single cell atlas of non-human primates, and these are some other members of my lab in Guangzhou. And with this, I would like to thank everybody for their attention. If you have any ideas of collaboration, please don't hesitate to contact us, and you can use this email address. Thank you.
Miguel, thank you very much. That was wonderful. I must admit, it's absolutely phenomenal to see those spatial slides of literally a whole organism on one image. That's quite remarkable. And your audio was great. You could hear everything perfectly. So we've got about 15 or 20 minutes left for a sort of interactive Q&A with the panelists. So can I invite the other panelists just to put their videos on? We've had a number of questions come through, both on the chat and a couple through Twitter, and a few from panelists that we'll sort of roll through. But I might also throw a few questions to between the panelists. But can I just start with a question to Miguel about the spatial work? Could you comment on what the computational challenges you face if you're generating data on slides of high resolution spatial sequencing of 13 centimeters squared? Did I read that right, or am I imagining that?
Yes, you read it right.
I was like looking for millimeters, but so how do you handle that amount of data in terms of some of the the memory implications?
Okay, well, it's quite of a challenge actually, and then there's two issues. One is how to store the information. So you probably know that BGI, well, we have a large capacity to store information, and we still haven't got to that level that we have been saturated. But we envisage that once we start sequencing a large-scale number of samples in a spatially resolved manner, we'll need to come up with a different solution. And then the solution that we have is putting information into DNA, and then sequence that DNA and basically store the principle of life on Earth would be the principle of storing information for videos, for anything, so including all this spatial information. So that is where we are heading to, and I think it's going to be quite revolutionary. We have achieved it. It's fantastic. The only thing is that we are trying to make it also cost affordable. But I think in several years from now, that would be the way of storing all the massive information. Then how to interpret? We're still developing algorithms because every organ is different. Every type of system, like understanding development is not the same as understanding disease. Depends on how big the section that you try to get deep into is. So we are developing algorithms so that we can have pipelines that can tell us everything simultaneously. It's going to take some time, and we're very happy to work with people from all over the world to develop new ways to get the maximum information from these sequence chips. So basically everything is in progress, but so far so good.
Yeah, remarkable. Thank you. So we've got a question in the chat, which is actually one that I had in my head and one that we've been sort of thinking about in the Gum Weissman Center as well. So I'll chuck it to Rachel first of all, but it's really around, we've seen a lot of methods being developed, or even methods developed, or the application of these combinations of short read, long read approaches using different types of single cell library prep. So we've had a question come through from Peter Van Galen, who's asking, is the only limitation of long read single cell data due to cost, or are there advantages if you do targeted limited sequencing? So imagine you were not in Australia and you potentially worked at BGI and money was no object. Would you just be sequencing as deeply as you are with your Illumina reads?
Probably. I think like now, because we're processing up to 10,000 cells on the 10x, like to get enough rates, we need to almost run 10 flow cells on the nanopore. And I think maybe people that know how much a flow cell is know how much that will cost in the end. But we did this subsampling approach because it's more an unbiased approach. And then we can just look, can we detect novel mutations? Can we detect novel isoforms? And if we see that and we can overlay that really nicely on our like short read sequencing, can we then we can always come back with like the more captured approach, because we still have, of course, this 10x full length cDNA stored, because you never use that much anyway for like library prep. So then you can always come back with a targeted approach. And I think for us, it works really well. We now see like novel things that's popping up, and then we can come back with a more targeted approach. So yeah, we don't have to run 10 flow cells. I think it's a little bit crazy to do that for like one sample, also cost wise. Maybe at BGI they will do it.
Miguel, would you do it? You do that at BGI?
You mean the long read?
Yeah. What would be the approach you would take in these scenarios? Would you just sequence the entire library deeply with a combination of technologies, or would you go down a targeted approach?
So we're doing long read also with the C4. It works, but obviously we cannot have so large scale. So I think it's a combination of the two things that would get us the whole thing. But we hope that in the very near future, we're going to be able to do large high throughput and also very cost effective long read single cell. So I think ultimately everything would go just do everything large scale the best way possible. But right now what I would suggest is, like we have been hearing, a combination of the two things, and you can get tremendous information if you do like that.
Yeah, thank you. Yeah, so Mike, you're addressing I phrase it, if you don't mind, as a particular biological question where you've got a really clear reason for needing to use a targeted based approach. And so can I ask what your view is of some of these methods where there's a targeted capture of the RNA as opposed to necessarily a capture of at the cDNA stage? So I'm thinking of systems like the Rhapsody and then some of the new panels that are coming through from 10x. Are there advantages for the types of biological questions you're interested in using those capture panels? And if there's not, what would you want to see to enable your research to progress with targets on the actual original RNA capture?
Sorry, do you mean is there an advantage of capturing the original RNA versus the cDNA?
Yeah.
It's going to depend on the type of library prep you do. If you've done a library prep where you effectively the cDNA is, the full length original RNA, then it's often easier probably to try and actually do the targeting on the cDNA. cDNA is tough; it doesn't degrade easily. It's easy to work with. I guess you can try and capture on original RNA. The issue being, of course, then you have to take all the extra precautions because you're dealing with that. We're dealing with, you know, RNA which is vulnerable to degradation. So, and you know, even if you're using a 10x type approach where the standard sequencing is you just go in for the 3', you can obviously adjust it slightly so that you do your capture before you do the fragmentation, and then you have like the whole cDNA to go at, just like this happens when you do the sort of nanopore long read split, where you split the library in two. So generally I would go for the cDNA because it's just easier to work with, and then you can pretty much target whatever you want.
Yeah, I think, my sort of personal feeling, there's a few applications that I can see where, you know, potentially a targeted capture, or particularly the application of those technologies. So if you think of clinical translation, where you potentially don't want the regulatory problems of having the ability to capture off target effects and incidental findings. But as Rachel pointed out, I mean, having those libraries that are, you know, not unexhaustible, but remain there, and you can go back and you can use different types of capture or different types of targeting to address different questions, sort of future proofs many questions that you might have, right?
Absolutely, those cDNA libraries will last in the fridge or freezer for years, and then if you've prepared them well, such that you maybe have a whole full-length cDNA in there, then you can effectively apply a whole gamut of technologies to it, be it long read sequencing, long read capture, short read capture, whatever you want.
Yeah, thank you. So we've had a question, another one come through from to Rachel from Jose Alcazar Hernandez, who asks, how are the CLL cells with the new isoform specifically tested for the effect of venetoclax?
So the new that's a cell line. So we knocked out that gene and then we expressed again the gene in it. So unfortunately we can't do much more like CRISPR editing in like primary CLL cells. So we took a cell line that's normally very sensitive to venetoclax, we knocked out that gene, and then we brought back the gene in like the normal transcript or like the novel one, and then test sensitivity to that. Yeah, unfortunately you can't do that yet with primary CLL cells. Yeah.
Thank you. So we have another one that's come through from to Miguel. How does the clustering analysis work for stereoseq? Are the spatial positions that you capture take into account, or do you do the clustering in a similar manner to you'd use with traditional single cell RNA approaches?
Okay, so sorry if I did not explain this clearly. So our DNA nanoballs are barcoded, especially barcoded. So then the chips are patterned with the DNA nanoballs. All those have a spatial information, and then what we do is we detach them and then we sequence and we know where they belong to. So I think this probably answers the question.
Yeah, thank you. There's also there's a couple of questions actually around just you might have covered this in your talk, Miguel, but I thought I saw some numbers, but someone's asking, Min Tan's asking if you're able to share the number of genes in the UMIs per.
Yeah.
Is spot the right word, or do we have a different word? Pixel.
Well, if I guess you can use any term. We call it yes, spot. Spot is fine. So well, this is evolving as we talk. Okay, we are improving it. So by per 100 micrometers, we can get a combined number of around 10,000 genes, and then the UMIs can be 30,000, 40,000, around that. It depends a little bit on the tissue. Some tissues are easier, some tissues are more difficult. As you can see there in the embryo, it's quite amazing the resolution that we can get. So and then per spot, and you can consider that every cell, a cell that could be like 20 micrometers could have around 400 nanoballs. Per spot, we can get 1, 2, sometimes more genes. But we are going to be, this means that per cell we could get 600, 700 genes, depends. And then we are aiming to improve this to a point that we would be able to have an even higher number of UMIs and even have a higher number of genes and it's going to work because actually the principle is so amazing and so simple in some way that there's no reason to believe that it cannot be enhanced.
Yeah, brilliant. Thank you. So I'm kind of keen to go back to some of the challenges that you have of integrating, short and long read data from a single cell library where you do a capture or a non-capture based method. And this is something we've struggled with, or we've been working on. I'm not so sure it's a huge struggle, but it certainly flags some concerns around the ability to match the patterns or the calling of the cell and UMI barcodes from nanopore to Illumina sequencing, and essentially then the dropout of reads that you're able to map through both sequencing. So, Rachel, starting with you, can I ask? I know you've worked, with Louis and Matt on this. So, and they might be online and can chat if this is in their wheelhouse. But has that impacted your experiments? Is this a challenge for you, or have you made any major advances in this space?
So I think Louis analyzed all our data, so we're really collaborating with Louis. And I think with Flames, what he developed, it is really good for the FLIT-seq. So we don't see, I think I didn't mention it, but for instance, the G11V, we didn't see that back in the T cell. So FLIT-seq is really good. We are having some challenges now with the RAGE-seq, and we're trying to change the pipeline a little bit for Flames, because it's of course newly developed for this long reads, short reads integration. How many reads we lose, I think we were able to, in the end, get around 40% good reads coverage back to what should be for this CLL cells. It's a little bit different for cell lines that I use, and I think it's all now should be available. We have a bioarchive. Louis has a bioarchive paper out there describing this. But yeah, I think Flames is really helpful that he made this pipeline for it.
Yeah, that's good to know, because I must admit I've not read the bioarchive paper, but I know there's people working on that very specific problem of mapping the calling of the barcodes, particularly at the start of the read from nanopore data, and that percentage drop is, as you said, that creates some potential challenges. So I think it's one of those areas. If those of you listening are really into this space, then I think there's probably some really exciting algorithm development that can come, because I see as you sort of heard from the talks today, that these combination approaches, I think, are just opening up so many biological questions. Mike, what's your perspective on this? Does this create challenges for your type of workflow, or something you sort of think about with your research?
Yeah, I mean, I think the basic method that you guys at the Garvin has used and Matt and Co have used, where you sequence you do the short reads to help identify the barcodes, and then that allows you to pull more reads out of your nanopore, works quite well at the moment. I think it will solve itself over time to a degree, because the accuracy of the nanopore sequencing gets better every year. And I would say probably in a few years, it will have largely resolved the issue just through better raw read accuracy. And I assume at the moment, if you're doing, for example, PacBio hi-fi sequencing kind of stuff, it won't be a huge problem there, because the accuracy is probably to the point where you can resolve most of the barcodes. It's also possible that could be solved kind of at the level of the barcodes themselves, if you eventually hack the system or use a system where you can just make the barcodes longer, and then a slightly more error-prone system won't have such a problem with them if the barcodes are 20 nucleotides instead of 8 or 10.
Yeah, thanks very much. So we're sort of drawing towards the end of this session, but I'm just going to put everyone on the spot, and so for those listening, I've not prepped our panelists this question. So if you're writing a letter to Santa of your wish list of what you would like for Christmas, what technological development or assay or method in the single cell or spatial genomics space would you wish for? And to give you a moment or two to answer that, I'll give you my perspective, because I've had slightly more time to think about this. So I really would like to see methodological development around linking individual cell genomic readouts to physiological components of the cell. And there's been some nice methods, fairly niche methods developed in this space. So things like single cell patch clamping, where you're able to generate the sequence the RNA per cell. And a lot of the questions we were interested in, that physiological readout, or let's call it cell phenotype, in addition to the omics of a cell, would be super valuable. So I'm going to start, that's my perspective, but Miguel, you're heavily involved in this space, so this might not be so much of a wish list, it's you're already doing it. So what would be something that would be fantastic for your research to see?
Well, like I said before, we are working actively on getting the single cell ATAC sequencing, the single cell, they especially resolve ATAC sequencing. So I hope we can complete these by Christmas or around Christmas, or straight after Christmas. So that would be like a dream come true. If we could get transcriptomics information, especially combined with epigenetic information, I think this would be an entire revolution.
You've been challenged to get it before the 25th of December now, right?
We'll try at least, in trial method. And like I've been saying in some of my slides, we're very happy to work with anybody from all over the world to improve what we have or give it a different angle. And then I think there's a huge room for epigenetics, especially resolve.
Yeah, thank you. Rachel, what's on your wish list?
I think the same, like epigenetics, single cell, more like maybe even single cell methylation, looking at that would be very interesting. I think also like on the long reads that you can more target it would be the next steps, I guess. Yeah, but also I think the same thing, epigenetics, combining good epigenetics with transcriptome would be really good.
Yeah, no, I suspect that's on a lot of people's Christmas. Mike, do you have anything that you feel that we're missing?
Well, my first thought was I quite like StereoSeq for Christmas, to be honest. Especially if we could do long reads with it.
There you go, Santa. Can you send him some? Danny, do you work in this space a lot? Do you have anything that you feel that you would like for your research, or something that you come across commonly in for people that you collaborate with?
Yeah, I think the spatial aspect is becoming like very high on everybody's list of demands, and I agree with Mike. Like I would also, if they're already shipping StereoSeq to Melbourne, I would also like one. And combining that really with that high resolution and potentially longer reads to really understand how differential splicing within like an organ can affect the way it functions, I think is a question that will be started to be tackled in the next year probably. So yeah, definitely on the top of my list.
Yeah, fantastic. All right, well, I'm making notes. So I'll pop that letter in the post and we'll see what we get. All right, so I think we're drawing right to the end. So I won't ask any more questions of the panelists, and I'll just sort of close up. I'd like to start by thanking our panelists for their time and sharing some really exciting work, and I found it absolutely fascinating. So thank you very much, and also appreciate the combination of the technical issues and the rapid rearrangement of the order. So thanks very much for your agility on that. Thank you, Danny, also, for co-hosting this episode with me, and Tom Ashurst, who is a black screen at the moment, who's been in the background helping us with AV for this episode as well. So thank you, Tom. I would also, of course, like to thank our sponsors for this episode, so Decode Science and BGI, and the sponsors for the whole series as a whole. And this, those of you who've been following the single cells journey this year, so we had a scramble with the onset of COVID to go from an in-person to an online conference. This is the 11th episode in a series of 12, so the final episode is next week. I'll both be sad and sorry to see the end of the 12 series episode. It's been a lot of work for the organising committee, so thank you very much to my colleagues that have been working on this. Next week's session is I think a little bit different, and I'm really looking forward to it. So we're having a combination of a panel discussion with these 4 panelists that you can see here, and there's going to be a particular focus not so much on biological ideas and technology ideas, but also looking at aspects of scientific careers through academia into industry, into publishing, and possibilities, particularly for ECRs and MCRs, of moving between different sectors and what the opportunities are, and what the landscape looks like. And so we're particularly excited to have Camilla and Barbara join us to offer their perspectives in this space. We'll also be announcing the winners of the poster competition. So the poster session is currently live. If you're really keen and rapid, there's another 5 hours available, I believe, to submit a poster, and these will be judged over the weekend, and there'll be some usual single cells nice cash prizes for the winners of this. And also, finally, keep an eye out on Twitter, because we'll be running over the next week the now long running for the last 4 or 5 years single cells Krisny Award. So for those of you not familiar, this is an award that I'm going to use the word commemorates Christine Wells's interpretive Thijssen dance at the first Oz Single Cells conference. And so the fact that it's a Krisny, a Thijssen award, gives you an idea of the age of the conference and when this came about. So there'll be some posts on Twitter to keep an eye out, but the idea is, as you submit any form of images or videos, or actually I believe any form of content to that Twitter account that's an interpretation of a Thijssen or UMAP or whatever we're using these days, and it will be judged by the panel, and also probably a popular vote. So keep an eye out on Twitter for that. So with that, I'll close up the session. Thank the panelists very much again, and thank you all that have been online listening for your attention and your time. Thank you. Bye.